Today's topic is the genetics of human disease. And so the questions we're going to talk about are what is a genetic disease, and how is genetic information being used to diagnose and to treat disease. Okay, so I want to take you back a little bit in history to the first identified genetic disease. This goes back to the year 1902. And so this is quite amazing, right? So if you remember the timeline of genetics, it was in sort of the mid-1800s that Mendel did his work, but people didn't really know about it until it was rediscovered at about the turn of the 20th century, so at around 1900. So the rise of human genetics coming at about the same time is kind of remarkable, that it was about the time that sort of Mendel's experiments were being rediscovered, and their application to humans was actually recognized fairly soon after by a guy named Archibald Garrod, who's a physician in London. And so what he recognized was that a particular disease called Alkaptonuria appeared in families in a way that was reminiscent of sort of genetic inheritance. Okay, until that time, until he made that observation, it was assumed that this disease, and we'll get into what this disease is in a second, was caused by an infectious agent like a bacterium or a virus or something infecting people. Which obviously, you know, it's looking back, it's easy to say, well, how foolish, why didn't they just recognize it was, you know, being inherited in families? But you have to realize that, you know, infectious diseases are probably also clustered in families as well. You're more likely to transmit an infectious disease to someone you live with, for example. And so it wasn't immediately obvious what the causes of such diseases were. And so he recognized that this particular disease appeared to show recessive inheritance, that his parents were unaffected and then children were affected, but then multiple children, say in the same family, were affected. Okay, so what is this disease? So if you recall back to like the very, very beginning of the course, when I told you how sort of DNA was first isolated, it was this guy Miescher who went and collected bandages from a hospital, realizing that they were full of white blood cells and had lots of nuclei in them, and he was trying to isolate the nuclei and study them chemically. So it wasn't a very sort of glamorous start of DNA research. The start of genetic disease research in humans was also kind of not so glamorous, as you can tell by the description of the disease, Alkaptonuria, that Garrod was studying. Okay? So the constant feature of this disease is the excretion of homogentisic acid, to the presence of which substance the special properties of alkaptonurin, the darkening with alkalis and on exposure to air, the power of staining fabrics deeply, and that of reducing metallic salts are alike. So sorry for the sort of Victorian language, but what that basically means is the disease is one where the urine turns black in exposure to air. It was usually recognized when diapers of babies had this black substance in them, and then various chemical tests could be done to sort of confirm that that's what was going on. So this is Garrod's paper where he reports his observation that it was probably due to a genetic inheritance in 1902. And one of the other things he says in this paper is that there are good reasons for thinking that Alkaptonuria is not the manifestation of a disease, that is not an infectious disease, but is rather of the nature of an alternative course of metabolism, harmless and usually congenital and lifelong. So alternative course of metabolism, he's sometimes also called an inborn error of metabolism. Okay, the idea being that you have it from birth and you have it your whole life. It's something different about your physiology, not anything to do with being infected by anything. Okay, the harmless part is not quite true. Okay, this homogentisic acid that builds up can damage bones and cartilage and lead to arthritis. It can also lead to cardiovascular disease because this black pigment is actually getting deposited in the body. But it's something people could live with and just would have this chronic illness their whole life. How did he recognize that it was actually a genetic disease as opposed to an infection? Well, this is what he said later on in the paper. The information available as to the incidence of alkaptonuria is of great interest in connection with the above view of its nature, that the peculiarity, that is the disease, is in the great majority of instances congenital, cannot be doubted, that is, people have it from birth. The abnormality is apt to make its appearance in 2 or more of the brothers and sisters whose parents are normal. Okay, so this is basically the hallmark of recessive inheritance, right? That you have 2 parents who are carriers of the alkaptonuria disease allele. They're both heterozygous. They have 1 good copy, 1 bad copy. Neither one of them has the disease. But when they have children, then those 2 bad copies of the gene can get together and cause the disease. And because the 2 parents are carriers, however many children they have, they're all subject to potentially inheriting those 2 bad copies, and therefore it tends to run in families. And you find brothers and sisters in the same family who are affected by the disease where the parents are normal. Okay. We now know that alkaptonuria is caused by a defective enzyme, and you've seen examples of changes in enzymes causing changes in phenotypes before. This is a simplified version of this sort of physiological pathway, the metabolic pathway, where this enzyme participates in the body. And so it has to do with the digestion of proteins. So we all eat proteins. The first step is to digest them down into the amino acids. There are 20 amino acids. One of them is tyrosine. Okay? And so the idea is that tyrosine can be used to make more proteins once it's in this form. Or, depending on what the needs are of the metabolism of the individual, and sometimes, for example, you have an excess of amino acids and you don't have enough carbohydrates, and so you would use these for energy instead of for building proteins. Okay? And there's this pathway that eventually converts tyrosine into energy through many, many enzymatic steps. Okay? And the step where this gene that's affected in alkaptonuria acts is in this step converting this homogentic acid into this other hard-to-say acid, which eventually gets converted, and makes energy for the cell. Okay? So the gene encodes an enzyme called homogentic acid oxidase, which does this conversion step. Okay? And that's why when people are affected by this disease, this enzyme is not functioning properly. So instead of going all the way through the pathway, you basically are blocked here. There's a roadblock in the pathway here. And so this builds up. Okay? And it's this substance that turns black on exposure to air and causes the phenotypes that are seen in the patients that have the disease. Okay? So thinking about it in this way helps us understand why this might be a recessive disease as opposed to a dominant disease. Okay? The idea being that if you have 2 bad copies of the gene, then you don't have this enzyme at all, and so you can't convert this into this. But if you have 1 bad copy and 1 good copy, you're still making some enzyme here. You're making half as much as other people. Okay? But that's enough, apparently, to keep converting this into this so that you get all the way through and you don't build up this. Okay? 1 bad copy of the gene is not terrible. 2 bad copies of the gene is what gives you the disease. Okay? So in addition to recognizing that alkaptonuria was probably a genetic disease, Garrett sort of developed a theory. He developed a sort of generalization of that idea and suggested that many human diseases might be genetically based as opposed to infections. This idea was at first controversial. Okay? Remember, it was the early days of genetics even. But even in his lifetime, it became sort of accepted that there were many human afflictions that were actually caused by bad genes. Okay, and that's something we know for sure is true today. And it's so true that we have huge databases that sort of summarize lots and lots and lots of research that's been done over the past 100 years or so to identify and characterize these genetic diseases. So in particular, there's a database called OMIM, which stands for Online Mendelian Inheritance in Man, that is very useful to people working in the field. And so the idea is that it catalogs all genes that have been shown to have some kind of association with human disease. And it's not just Mendelian diseases. Usually when we say Mendelian, we mean sort of 1 gene, 1 disease. But it also covers complex traits like heart disease, diabetes, and so on that are affected by more than 1 gene. And don't show strict Mendelian inheritance where you could say, aha, it's recessive or it's dominant. but actually are due to many, many factors interacting. So anyway, this database is huge, okay? And so we can look at sort of a crude characterization of what's in the database. The number of genes where there's a Mendelian disease, that is a disease caused by a change in a single gene, with a known mutation where, that mutation that causes the disease is actually known, okay, is about 350 and growing, okay? So if you think about the genetic diseases that are Mendelian, they're things like Huntington's disease, cystic fibrosis, Alkaptonuria. So these are the kinds of things that fall into the 350. Lots of ways single genes can go wrong and cause human disease, okay? But there's still a lot left to discover. So also in the database are about 1,800 diseases that show Mendelian inheritance. That is, by looking at pedigrees of families, it's clear that there's a bad version of the gene that causes the disease, and it's basically one gene that's causing the problem. But that gene is not known. That is, the mutation in that gene has not yet been identified, okay? So there's about 1,800 to go that people are, to different extents, still studying to try and figure that out, okay? And then, as I said, there are lots of diseases or human conditions that are not due to just one gene, but many genes potentially can affect their expression. And so the database also contains a large number of those types of diseases, 2,689 in fact, where there's more than one gene that's associated with those diseases, okay? And so I just want to show you real quick what the database looks like. It tends to be fairly technical information because it's meant for researchers who are active in the field. It's not necessarily meant for the public. But we can just sort of get a sense of what's in it by doing a search for something like Huntington's disease. Okay? And so if we click on what's found here, right, you'll see the kind of information that's in this database. So here we have Huntington's disease. And if we look at the description, it says Huntington's disease is an autosomal dominant progressive neurodegenerative disorder. We've already talked about all of that it just takes one bad copy of a Huntington's disease gene to have the disease. Characterized by various things, basically neurological dysfunction, loss of coordination, and eventual death. And so it gives a description of the clinical features. That is, if someone comes into a hospital with certain symptoms, these are the features that are used to identify this particular disease. And you can see there's a lot of research on that. Different research on juvenile onset, so children presenting with the disease. And as you know, it's more likely for a child to present with the disease if they have more copies of that repeat that's in the middle of the Huntington gene. Biochemical features. So physiological aspects of the cells that are affected when someone has the disease, and inheritance, so autosomal dominant, how it actually works, and so on. Okay, and you can see there's tons and tons of information on stuff like that. Okay, so that's basically the database. You can look up other things on there, cystic fibrosis and so on. It's all in there. And also, I should say, if you look up something like diabetes, okay, you'll get a lot of entries, so 746 entries of something having to do with diabetes. So that's a complex trait with many manifestations and also many genes that are potentially causative factors in it. Okay. As you saw, there were about 350 genetic diseases where the genetic cause is actually known down to the molecular detail. There are about 1,800 more that are still left to figure out, and they fall into various categories. We've talked about sort of these neurological disorders that are associated with these trinucleotide repeat genes, but this is just a summary of genes and disease from an online book by the National Institutes of Health, and you can see by the categories that there are many, many, many types of disease that one could get due to a problem with the genes. Okay, so in addition to sort of muscle and bone diseases, nervous system diseases, respiratory diseases, diseases of the eye, diseases of the digestive system, diseases of the blood, male-specific, female-specific diseases, ear, nose, and throat diseases, cancers, and so on. So there are many, many, many different ways your genes can go wrong and cause disease. Some of these diseases are exquisitely rare. Okay, some of these conditions don't occur very frequently at all. And as you'll see, especially for recessive conditions, we have some expectation that they might be rare, except in some unusual cases. But some of them are actually quite common. All right, so here are some of the most common human genetic diseases. Okay, and one of the things you'll see is that they're common, but they usually tend to be more common in certain groups of people. So depending on their ancestry. Okay, so sickle cell anemia. This is the carrier frequency. So that's the frequency of heterozygotes in the population. People who, if they had children with another heterozygote, could have offspring that are affected, right? Okay. So this is not people with the disease. This is people who have the potential to have children with the disease, right? Sickle cell anemia. 1 in 10 African Americans is a carrier for a sickle cell anemia, bad version of the gene. Cystic fibrosis. 1 in 20 Caucasians is a carrier for that. And Tay-Sachs disease, 1 in 25, and it has an unusual distribution where it's very common in Eastern European Jews and French Canadians. Okay, and so these are really, really common. Okay, if you think about sort of this room, you know, there are certainly carriers for one or more of these diseases just in this room, certainly, no question about it. As I said, for recessive diseases, even if the carrier frequency is fairly high, the number of affected individuals is going to be lower, and that's because it takes 2 bad copies of the gene to be affected. Okay, so one way to look at that is if you have a simple pedigree where you have a father and a mother and a child, and so the father is a carrier, so 1 good copy of the disease gene, 1 bad copy. So let's say this is Tay-Sachs disease, so good copy, bad copy, good copy, bad copy, and then the offspring might get the 2 bad copies. What's the probability that this offspring, given what these parents are, what's the probability that, say, another child from these parents is going to have Tay-Sachs disease? Yeah. 1/4, right? Okay, and the reason it's 1/4 is there's a 50% chance that this parent gives that allele. Okay, and there's a 50% chance that this parent gives that allele, and so 50% times 50% is 25%, or 1/2 times 1/2 is 1/4. Okay, so the probability of this child being affected, given that the parents are carriers, is 1 in 4. Okay, now, as I said, what we can now do is think about it in terms of not that we know what the parents are, but we can add in the probabilities that these 2 parents are carriers or not. Okay, so let's imagine that we didn't know what the genotypes of the parents are. All we know is that they're not affected. Okay, and we know for a particular disease gene, we know what the probability is that those parents are going to be affected. Okay, so for Tay-Sachs disease, I said, let's say these are Eastern European Jewish parents, and each one of them has a 1 in 25 chance of being a carrier. Okay, so that's 4%. Right? So given that carrier frequency, we can estimate what the probability is in general, just in that population, of getting the disease. Okay, so the way that works is not only do these 2 alleles have to be passed on to the child, that's the 1/4 probability, but there's also a 4% probability that this parent is a carrier and a 4% probability that this parent is a carrier. Okay, and if either one of these parents is not a carrier, there's no way this child can get the disease, right? They could be a carrier again. And so the way we figure out sort of the population frequency approximately of the disease is we take 50% times 50%, and we also multiply 4% and 4%, okay? And so that's what's down here. We have 4%, 4%, 50%, 50%. And so that gives you 0.04%, or 1 in 2,500, okay? So this is really interesting, right? So if you just look at the carrier frequency, it's a 1 in 25 probability of being a carrier, okay? But the probability of actually having the disease is much less. It's 1 in 2,500, okay? The more rare a disease is, so the more rare the carrier frequency, the lower the carrier frequency, the bigger the difference there is between the carrier frequency and the incidence in the general population. That is, for common diseases like Tay-Sachs, this number is indeed smaller than the carrier frequency, but it's not too small. I mean, 1 in 2,500, okay? And if you think of, you know, sort of all the people with French Canadian or Eastern European Jewish ancestry, that's potentially a lot of people that would end up being affected by this disease, okay? If we had a much more rare disease, this number would be that much lower. And it's not proportionally lower, but very, 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 very low, okay? So the way to think about it is that for diseases that are really rare, that are genetic, they're almost always caused by relatives having children with each other, okay? And that's because the carrier frequency is very low, okay? And the way a person gets two bad alleles of something is not by two random people from the population having children, okay? Because then these probabilities are much lower than 4% for a very rare disease allele, okay? And so usually the way it works is whoever is affected actually has parents who are related to one another and inherited the bad allele from the same person in a higher generation in their family, okay? Yeah, so this is an approximation, but it's a very good approximation. Okay, so it's a good rule of thumb that you can use, and you could always use it. So if I told you, you know, let's say I wanted you to calculate the incidence of sickle cell anemia among African Americans, and I told you what the carrier frequency is, it's 1 in 10, then you can use the same formula basically to get it. It would be 10%, 10%, 50%. Okay, so back to this number. As I said, you know, for a common genetic disease, this is a fairly big number, right? And so various things have been done about it over the years, and in particular, in some ethnic populations, there's been pressure to actually find out if you're a carrier before your potential mate and you get together and have children, okay? And so here's an article from a few years ago basically declaring Tay-Sachs, which historically has been known as a Jewish disease just because the carrier frequency is so high among Eastern European Jews, being eradicated, okay? So that sort of goes against this number of 1 in 2,500 that I just told you. And the way it's achieved basically is that it's very common for two people of Eastern European Jewish descent to actually get genetically tested before they have children to find out if they're both carriers. Because if they're both carriers, they have a 25% chance of having a child with the disease, okay? And that testing has been so extensive that basically very, 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 very few babies of two Eastern European Jewish parents get Tay-Sachs disease because it just doesn't happen that the two parents would actually have children together. Okay? And so in 2005, for example, not a single Jewish baby throughout North America was born with Tay-Sachs. Okay? And so if you compare that to the probability of a baby being born with Tay-Sachs, if there wasn't this kind of effort going on, that's a huge, huge difference. Okay? And there would even be some advantage to sort of expanding the screening to everyone, right? Because even though there were no Jewish babies with Tay-Sachs in that year, there were 10 babies born in North America with Tay-Sachs, but not a single one of them was Jewish. Okay, so that was just sort of bad luck. That was the bad luck side of things, where in some other populations, it's a rare allele, but two of them got together and one of the children. Okay? And so there's a lot of this kind of genetic testing that goes on. And in fact, if you go up to NYU Medical School, in the hospital, there are lots and lots of tests that they do every day. And so they have this whole web page on carrier testing. Okay? The idea being that if you're planning on having children, you can find out if you're at risk for, you know, the two parents having basically the same bad disease gene. Okay? And if you click on this link here, you can see all of the things they test people for. And I want to see, it might not show up that well on here because it's such a tiny chart. So I'm going to try and, let's see if I can just bring it up through the website. Right, so they have this brochure for genetic testing at NYU School of Medicine, okay, and then they have this chart with all these diseases that they test for, okay? And I'm going to read it out because it's tiny. But these are the diseases, okay? So this is a list of about, I don't know, 12, 15 diseases. These are the signs and symptoms of the disease, the outcome if you have the disease. I don't know how well you can read it, but that word there is fatal. That word there is fatal. That says early death. So these are very, very serious conditions that are either extremely debilitating if people do live, or they actually cause very early death. The other sort of depressing column is this one, which says possibility for treatment. Okay, and so that says none, 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 none. Okay, so in those cases, basically the only way to avoid this very terrible condition is to make sure it doesn't happen in the first place. Okay, because once someone has the two bad copies of the gene, that's it. They have it. There's no hope for any kind of cure or treatment. This lists the carrier frequency and which populations they tend to be high in. And this lists the type of test that's done. The first word of every one of these is DNA, basically. So they get DNA samples from the people, and then they test for the mutations that are known to cause the disease. Okay? So one of the really interesting parts of this table is, so like Tay-Sachs is on here, right? Okay? Rapidly progressive disease, fatal by age 4 to 5, no treatment for it. Frequency of 1 in 25 in Jews of Eastern European descent and French Canadians. 1 in 300 in other groups. Okay, you can see what the difference is there between certain groups having a disease and certain not. If you look down this list, there's 1 in 80 in Jews of Eastern European descent, 1 in 40 in Jews of Eastern European descent, 1 in 30 Jews of Eastern European. So talk about being like a chosen people. Okay, this is like really, really kind of depressing news for some of us who are of Eastern European Jewish descent. Why do you think that is? Why would sort of a group like that, and there are other groups too, right? French Canadians are on here, African Americans in some cases, right? So why would certain groups be more prone to disease than other groups? Yeah, so that's exactly right. So within that group, there's less genetic variation, okay? And the reason is that there are what are called founder effects. So the idea being that at a certain point in history, that group was really small, okay? And then the group might have expanded, and so there are more people today that are part of that group than there were ancestrally. And if there's a very small number of people ancestrally in that group that gave rise to everybody living today, then whatever sort of genetic baggage those ancestors had is going to end up in the gene pool, and that's the right word, of that group later on. Now, there's two conditions that have to be met for that to happen. One is that that bottleneck, that sort of restriction in the population to a small number, has to be pretty severe, okay? And the other is that intermating within that group has to be extraordinarily common, and mating outside the group has to be very uncommon, okay? That is, if people of Eastern European Jewish descent only have children with people of Eastern European Jewish descent, and that maintains that as sort of a small gene pool, okay? And if people don't sort of obey those sort of commandments of their parents to marry someone Jewish or whatever, right, then that gene pool just sort of mixes with the general gene pool, and there's no more sort of restriction to that one population in terms of these bad disease alleles. So this is the other very excellent point, which is that there might be some bias in terms of what's studied. Okay, and I'm going to show you an example in a second of efforts to sort of remove that bias. But it is certainly true that Jews are overrepresented and have been for a while in medicine, in scientific research, and have had practices in place, as I said, for dealing with things like Tay-Sachs disease even before DNA tests existed. So it used to be, you got interrogated by someone saying, do you have any cases in your family going back however many generations of Tay-Sachs disease? If you do, we're not going to set you up with her because she also has that, right? And so matchmaking was actually matchmaking. It was, not just how good of a sort of personality fit or whatever you were, but it was, do you have some possibility of having these disease genes in your family, and then stay away from that other family. Okay, and you can think about sort of the ethics of that, right? Sort of whose choice is it to do that? What options do people have when they find that they are a carrier, and so on? It's not clear cut, and I'm just sort of telling you what sort of the practices are. And it goes to this question of why, most of the things on this list are diseases that affect Jews of European descent. One is that there was a bottleneck in the population, so there is a limited gene pool, so it meets that criterion where there can be these kind of disease alleles. But there are plenty of other ethnic groups that also are like that. But the research has not been done as much in those groups, okay? And so that brings us to this example right here. So here's a disease, okay? It's kind of unusual. It has dominant inheritance. It's caused by the expansion of a triplet repeat of a kind, of Huntington's disease and so on. So like Huntington's disease, a small number of repeats is normal, and then a larger number of repeats leads to the disease. It's not quite as debilitating as Huntington's disease, but it is a serious condition. So the outward signs of the disease are sort of drooping eyelids, and this is a picture from the New York Times a few years ago. Muscle weakness, and the most debilitating part is difficulty swallowing. Okay? And so in this case, this is a disease that's found predominantly among French Canadians. Again, French Canadians start with sort of a small population, right? You have a settling of a particular place, and then sort of only sort of mating within that group for a while. And that, again, causes the same thing, where certain disease genes get mixed into that gene pool and then become sort of high frequency. So French Canadians and also people of Hispanic heritage. And what's interesting about this, and why it was reported a few years ago, is that through the DNA analysis, you can actually, and combined with historical analysis, you can actually identify where that bottleneck event happened in this case. Okay? And this particular disease can be traced back to 3 French sisters who came to Canada in 1648. Okay, that's quite remarkable combination of sort of genetic analysis and historical analysis to pinpoint basically the origin of a disease. And this disease does not affect a large number of people, and it affects a population that's sort of not usually represented on that list of groups that historically have had things to genetically test for. But it's one of the examples of trying to sort of make up for that disparity and get other rare genetic disorders that are limited to particular groups on the map so that people can get tested for them. I should say that the surest way to sort of avoid these kind of genetic diseases that are limited to particular ethnic groups is to outbreed, okay? To put it crudely, do the opposite of what all my relatives said to do, which is, marry someone of your own kind, right? The best strategy is marry someone not of your own kind, and then you can usually avoid all of these things. principle. So there's another issue that we'll talk about in a second that complicates it. But yes, certainly if everyone knew their entire genome, right, and you knew all of the causes of genetic disease, then you can just sort of, line it up and say, do you have any of these same alleles as I do? If no, proceed, have a family. If yes, then there's some risk there. Genetic testing has become enormously widespread. Okay, so this is a chart showing through the years the number of laboratories that do genetic testing in purple. So these are not like research laboratories like upstairs. These are laboratories where you send them the samples, they give you your test result. And the number of diseases for which testing is available. Okay, and so you can see that this is just sort of growing and growing and shows no sign of stopping. We're now at about 1,600 genetic tests now that can be done. Okay, this is your typical economic curve, right? Even though this keeps growing, consolidation of economic power happens, right? So that larger and larger firms are the ones doing all the testing. To give you some idea of sort of how widespread it is in the US, large proportion of newborns in the US are screened for one or more metabolic disorders at least. Not necessarily genetic disorders, because those tests tend to be more expensive, as you'll see in a second. But there are lots of tests for things like PKU, which we talked about in an earlier lecture, which can be treated, and so that's why these tend to be sort of widespread screening. For example, if a newborn is found to have PKU, they go on an altered diet right away, and that basically can cause pretty much all of the usual symptoms of the disease to go away. So about 4 million newborns per year are screened for a metabolic disorder. And this sort of gets back to what I was going to say about your question, which is that about 1,000 of those 4 million will be diagnosed with a metabolic disorder, okay? And about 12,000 will receive a false positive result, okay? So that makes it sound like these tests are really bad, right? That, why is it that 1,000 are correctly identified, but 12,000 are incorrectly identified? But it turns out most of these tests are really, really accurate. And the reason why this number is so large is because this number is so large, right? So if you think about it, among those 4 million, there are only about 1,000 that actually have the disease. That leaves pretty much 4 million that don't have the disease, okay? And so if you test that many people, 4 million people, for something, even if your test is really, really accurate, it's going to have some false positives. And so the result is that you end up usually getting more false positives than you get true positives. So 12,000 people incorrectly diagnosed and 1,000 people correctly diagnosed. So this is one of the complications that makes it difficult to screen an entire population for every possible thing that could go wrong, because you end up scaring a lot of people who have no reason to be scared. And you end up spending a lot of money sort of doing the testing and retesting, so it becomes sort of economically challenging too. In addition to all these Mendelian diseases, which have sort of simple genetic causes, one gene goes wrong, there are a lot of complex diseases that have multiple genetic and environmental influences. And actually, these are more common than simple ones, as you saw in that table that I showed from the OMIN database. So it's been estimated that less than 5% of non-infectious diseases, that is genetic diseases, are caused by a single gene. And in such a way that sort of the environment may affect sort of the severity of some symptoms, but it's not, in medical treatment it might, but it's not going to make that thing go away, right? And so that leaves more than 95% of genetic diseases being something where multiple genes and multiple environmental influences have something to do with it. And these are things like the very common human afflictions like cardiovascular disease, like cancer, like diabetes, and so on. And usually when we talk about these kind of genes affecting disease, really all we can say is that alleles of particular genes confer an increased or decreased risk of developing the disease. They're not an absolute thing. They're just saying that you're more or less likely to have a certain disease. And so this becomes a little bit tricky in terms of diagnosis, right? You can still get a genetic test for one of these, right? if you think about it, let's say I told you that I can test you for a certain gene, and if you have one particular allele, you have 5% greater chance of being diabetic, and you have another allele, you have 5% less chance of being diabetic. It's hard to know what to do with that information, right? If I told you, well, you have a 100% chance of getting Huntington's disease, you still might not know exactly how to process that, but it's clear what the outcome is going to be. Here, it's not even clear what the outcome is going to be if you have a 5% higher risk for something, okay? And so this presents a challenge in the clinical setting when people are actually getting tested. You know, what do genetic counselors tell people when they get these results? It's causing a really big problem that we'll talk more about, which is that there are now, you know, direct-to-consumer genetic tests where you can get tested for any, you know, thousands of different genetic features of your genome, and pretty soon we'll be able, for a small fee, to get your entire genome sequence, and then you'll have all the information there. But the challenge still remains, well, what do you do with that information? You know, how do you process all of these slightly increased or decreased relative risks for things? Okay? So I want to talk about one example where there is a real risk associated with particular alleles. It's not absolute, okay? And so it's a good sort of test case to think about in terms of what you do with this information. And it's a good example because it's actually been in the news this week. And that is these two genes that are associated with risk for breast cancer. And they're called BRCA1 and BRCA2, which is just short for breast cancer 1 and breast cancer 2. Okay? And so some of these statistics are really depressing too, right? So 13% of women in the general population will develop breast cancer during their lifetime. Okay, that's a really, really huge proportion. And that risk goes way up if you have the bad version of either one of these genes, because it's been estimated somewhere between 36% and 85% of women with a mutation in one of those genes will develop breast cancer. And it also tends to make it happen earlier in life as opposed to later in life. Okay? These are inherited as dominant disease alleles, so it just takes one bad copy of either one of these genes to increase your risk. Another way of saying that is that the carrier frequency is identical to the disease frequency, right? The carrier frequency is the same as being at this increased risk for the disease. Similar rates apply to ovarian cancer, which has related causes to breast cancer. Okay, ovarian cancer is more rare; it's only about 2% in the general population. But with a mutated allele of either this gene or this gene, it goes up to 16 to 60%. Okay, so as I said, these relative risks are really high. Okay, most genetic conditions will not have this kind of a stark contrast between the risk with a particular allele and without. But they're not absolute, okay? So, you know, there's a reasonably high proportion of women with a bad allele of this or a bad allele of this who will survive to old age and never develop breast or ovarian cancer. But from the data that exists now, most actually will not. Most will have to deal with breast or ovarian cancer. As with other genetic diseases, these bad alleles of these two genes tend to run in certain populations more frequently than others. And yet again, about 2% of Jews of Eastern European descent have mutant alleles, whereas 5 times fewer in the general population do. Okay. No, it's 50% passed on. Yeah. So right. So basically what it says is, you know, the carrier is heterozygous and the person who's at risk for the disease is heterozygous. So if I tell you what the carrier frequency is, it's identical to the frequency of people at risk, at higher risk. And if you have a parent that's heterozygous, then your probability of inheriting that one bad allele is 50%. Okay. So in this case, it turns out that nobody is homozygous recessive for this because presumably it leads to very, very, very early sort of abortion in utero, so natural abortion. Okay, so basically, you need this gene to survive, and if you have one bad copy of the gene, you can survive, but it puts you at high risk for the disease. If you get two bad copies of the gene, you actually never develop at a very, very, very young age. Not even age, very, very early stage in embryonic development. The embryo would not develop. And that's common of a lot of human genes. It's actually been estimated, so you might not realize this, but the rate of sort of spontaneous termination of pregnancy is very high, okay? And it's been estimated that each one of us carries somewhere around 8 genes where if they were homozygous, they would be incompatible with development, okay? So we're all carriers for, you know, some handful of genes that are that bad, that it's not even a genetic disease, it's just impossible for an embryo to develop if you have 2 copies of that. And so this is one of those which also, as a heterozygote, has a phenotype of increased risk for disease. So as you might imagine, it's more challenging to find these genes like this, that only give you an increased risk of something, as opposed to a gene that gives you 100% risk of something, right? So it's easier to find the gene that causes cystic fibrosis or causes Huntington's disease than it is to find the gene that causes a 60% increased risk of something, because for any individual person, you don't know whether they got breast cancer because they had the bad allele, or they didn't get breast cancer because they didn't have the bad allele. You have this sort of categorization error that goes along with not knowing until you found the actual gene whether any one person carries the bad allele or not, because there's some doubt what the cause of that person's actual disease or lack of disease is. Okay, so it took an enormous effort to actually identify these genes, and the way it was done was sort of similar to the kinds of ways we've discussed about how you find genes that affect traits. Okay, so these genes were, and I'll show you some clips in a second that explain this some more, but they started with families that had really high incidence of breast cancer. Okay, so within a family, many, many women affected by breast cancer. Okay, and then they tried to get families where they could get DNA samples from as many members of that family as possible, and they used molecular markers all along the chromosomes, and they tried to find molecular markers that correlated with the incidence of disease. Okay, and it would never be a perfect correlation, because some people with the wrong gene will still not get cancer, and some people with the good gene will still get cancer. Okay, so you have to use lots and lots of people, lots and lots of molecular markers to try and zone in on these genes, and eventually it was done by a sort of heroic effort by a heroic scientist named Mary Claire King and her colleagues. And so I just want to play. There's some correlation between these genes and cancer in men, although not quite as strong as cancer in women. And so the question is, well, so you go, and in this case, you go to Mount Sinai and you get tested for, BRCA1 alleles, BRCA2 alleles. They give you a result. What do you do with that test result? because, as I said, testing positive for one of the alleles does not necessarily mean that you'll develop cancer. Okay, there's some higher chance of developing cancer, but it's not absolute. But it does sort of mean that people should consider some options in terms of what to do. Okay, and these are sort of the most common things that people consider when they get a positive test result for one of these genes. Okay, and so one of those is just greater surveillance. Okay, so getting mammograms more often, so that if you do develop breast cancer, you catch it really early. Okay. Another is prophylactic surgery. That is surgery that's not sort of necessary to remove some cancer at the time, but is done to prevent that organ from actually developing cancer in the future. Okay. And so one common thing that's done is for women who are past reproductive age, it's recommended that they actually have a hysterectomy and have their ovaries removed if they have one of these two alleles, because there's a very high risk of developing ovarian cancer if you have one of those two alleles. And ovarian cancer tends to be extremely hard to detect early and very, very lethal when it actually occurs. Okay, so when my mother, for example, got her test result back and found that she had the BRCA1 allele, she actually had her ovaries removed, and they discovered very early stage ovarian cancer there. And so it's pretty much certain that her life is now sort of long and happy because of this genetic test that allowed that to be found and taken care of. Lifestyle adjustment. So sometimes, in cases like this, like cancer, there are other contributing factors that increase rates of cancer, right? And so one way people deal with having an elevated risk that they can't really control is to deal with the risks that they can control. It's sort of impetus to quit smoking and drink less alcohol, for example. And then the other thing that's available in some cases is that you can actually get drug therapy even without having a disease as a way of trying to, again, prevent the disease from developing in the first place. Okay? And so what I want to do now is actually play a clip from the New York Times Online, which had a really nice story a few years ago profiling a woman who had gotten a diagnosis, had dealt with a lot of women in her family having breast cancer, and sort of what she did with that information.
Not everybody agrees with the decision to do this. Not everyone thinks it's a good idea.
In 2 hours, Deborah Lindner will have a bilateral mastectomy to remove both of her breasts, even though she does not have cancer.
Mom, am I forgetting anything? Toothbrushes, hairbrushes, major surgery, and nobody wants to go through major surgery. Moisturizer.
But for 33-year-old Deb, surgery seems like the best strategy for a longer life. She is part of a group of healthy women genetically predisposed to developing breast cancer, a previvor.
Okay. I have some moisturizer.
When Deb was 18, her mother Joan was diagnosed with breast cancer.
I remember her being sick and tired and throwing up and laying in her bedroom during the chemo, and it was hard. It was a really hard time.
My first encounter with cancer, or the idea of it being in our family, I was probably too small to understand the word when I began hearing about it, because my aunts had it, my grandmother had died of it, and her mother.
With DNA testing, families can determine who has inherited the defective gene and who has been spared.
So this is my grandma Eleanor, and she's the one who had ovarian cancer shortly after my mom had breast cancer.
Mutations in 2 genes, BRCA1 and BRCA2, have been linked to breast cancer, raising the risk for carriers to as high as 87%.
So my sister is negative, and so obviously her children also are negative, so that's great.
But Deb, then a 32-year-old medical resident, received the opposite news. She tested positive for a BRCA1 mutation.
It was really tough to process the fact that this was something that was personal that I was going to have to deal with.
She began by scheduling frequent mammograms and MRIs, but she lived in constant dread of one day finding the lump in her breasts. After a year of researching and soul-searching, she called her mother with a plan.
She had come to the point when she needed to face this head-on, and that she had decided to go ahead with planning for the prophylactic mastectomy.
She was probably the most questioning, and are you really sure that you want to do that? And are you sure you want to do it now?
Is it not possible to prolong this a little longer, give herself a chance to find that special person, to be married, to have her family, to nurse her babies?
And I asked her if she would go back through my family tree and find out the ages with which people were diagnosed with breast cancer.
And when I went back through the family tree, I realized one of my cousins was 33 when she had her first bout with breast cancer. Deb is 33.
She came back a couple days later. She called me and she said, please go ahead and do the mastectomy. Please do it now. So I think she just really wanted me to take care of it. So
Next, Deb had to tell the man she'd only recently started dating.
obviously, when you've only been with someone for a few months, it doesn't seem appropriate to be talking about the most intimate things that are going on in your life. And I wondered what the response would be. And my aunt said, you know, if the guy's worth being with, he's really not going to care about that. He's going to care much more about you and about your health.
Baby, do Ice Age. That's cute.
Deb also faced the same body image concerns as survivors.
The scarring from many of the pictures I saw was just really not pleasant.
She opted for a form of reconstructive surgery not often performed in the United States that would leave her nipples intact and minimize scarring.
I don't expect to have it look natural, to have it look perfect.
That's good enough?
Yeah. Even though it brings up a lot of challenges and a lot of heartbreaking realities, it is such an incredible opportunity to know your risk. And it's really empowering to know the risk and to take action against it.
The mastectomy and reconstructive surgeries last over 7 hours.
So this is it's a little lumpy. That's going to go down, hopefully. These drains come out each side, and then that goes all the way down to here.
Is it painful?
Not yet.
But the pain does come.
Oh, stop. Ooh.
Deb remains in the hospital for 3 days.
Just did it from home.
She is cheered up by a special visitor.
Yeah, you're welcome to open your car if you'd like.
He definitely wanted to be supportive, and he wanted to be there as much as he could to support me through the procedure. It's really nice. Thank you.
Yeah.
Thank you for coming.
The surgery has almost completely eliminated Deb's risk of developing breast cancer. She will not be able to nurse, and she will have little sensation in her reconstructed breasts. But she has no regrets.
I'm really relieved. I'm just so happy to have this done, and I think overall I just feel really happy that I don't have to worry about this anymore.
So there you have it. Sort of pretty serious stuff when you get one of these diagnoses. The thing you saw at the very end, the text is kind of telling too, that it's estimated that there are many, many carriers and then only 30,000 will get tested. One of the reasons for that is that the test is very expensive and usually not covered by insurance, so it's $3,000 currently to get tested. And that's the reason it's actually been in the news this week. I don't know if anyone caught the stories this week about it. There was a lawsuit to sort of challenge the patent of a company called Myriad that does these tests because basically what they had patented was a gene, right? And usually you can't patent natural products. Like you couldn't patent your foot, you couldn't patent a rock out in the park. You can only patent things that are true inventions, creations. And so a lawsuit was waged to challenge the patent that Myriad had. And Myriad is not unique in having these kind of patents. On the Mary Claire King clips, you'll see the last two are called Patenting Chaos and Commercial Patents, and I encourage you to look at those. One of the things she says in that clip is that if you take any particular piece of DNA in the human genome, it's probably been patented 3 times over by now. Okay? So patenting is just sort of running rampant. And one of the effects it has is to lead to increased prices for genetic tests like this. And so a lawsuit was brought by a number of groups to try and break that patent, and the decision was actually a surprise to everyone. The judge decided in that case on Monday, I think, to declare the patent invalid. Okay, so that's not the end of the story. There will probably be a lot of, there will be an appeal. I would suspect that it would go all the way to the Supreme Court because it gets to the core of what can and cannot be patented. But for now, at least, there's some hope that the loss of the patent will actually lead to a much more affordable test because it will allow competition and other people to come in and charge less for that test. And if you read some of the news stories, one of the people who's interviewed a lot for this is Mary Claire King, and one of the things she says is that she's very hopeful about this because it will open up this kind of testing to more people who actually need it. Okay. The other thing that really needs to be said about genetic testing is in very, very, very few cases does a genetic test actually lead to the ability to have some kind of cure or treatment. Okay? In many cases, what we really are dealing with now is a genetic test that tells you you have some risk of something, but given that risk, it really doesn't lead to any sort of instant treatment or cure. There are a few now, but there's one sort of very, very important exception to that that's become like a classic example of how genetic information might actually lead to a next generation of medicines. They're sometimes called personalized medicine or genomic medicine. The idea being that if you know something about the person's DNA who has a particular disease, you're better able to treat that disease. And the example that's sort of the classic example now is a drug called Gleevec. And what that drug is for is a kind of leukemia called CML, chronic myelogenous leukemia. And this is caused not by a mutation in a single gene, but by a rare combining of chromosomes, so that chromosome 9 and chromosome 22, which are usually separate things, they break and rejoin with each other, which is not supposed to happen. And they do it in such a way that this gene here called ABL and this gene here called BCR actually get combined into a new gene. So this is a very sort of rare type of mutation that can happen where the chromosomes actually sort of change places and 2 genes get fused together. But it's not as rare as you might think because for whatever reason, these 2 parts of the chromosome like to break and rejoin with each other more than random parts of the chromosome. And so this is a mutation that actually happens during someone's lifetime. So that you start out with normal chromosomes, but then at some point as your blood cells are dividing, these chromosomes break and rejoin with each other, creating this fusion of these genes, and that's what leads to leukemia. So this is a really unusual situation, and what makes it cause leukemia is that this combination, this new gene, creates a protein that's normally not seen in the human body because it's part of one protein and part of another protein put together. And it just so happens that when you put those 2 parts of those 2 proteins together, your white blood cells divide without stopping. Okay, so they just overproliferate and they cause this leukemia. Okay, now because you have this very unusual situation where two genes have fused, that gives you obviously the potential to do a genetic test, because you can say, well, does this person have these two genes fused to one another or not? Okay, and it also led to the development of a drug that specifically targets that fusion protein. Because the way to think about it is that this protein, this BRCAI protein, only exists in people who have this particular type of leukemia. It doesn't exist in anyone who doesn't have that leukemia. Okay? And so what you can do is hopefully create a drug that targets this new protein, right? And that will have no effect on the normal BCR protein. It'll have no effect on the normal ABL protein, but it'll only inactivate the fusion protein. And then if you do that, then potentially you can cure the cancer, because even though that fusion protein can be made, it doesn't cause overproliferation of cells. And that's exactly what was done. So there's a drug now called Gleevec that was designed specifically in a rational way to target these proteins. Okay? And it does it really, really effectively. Okay? So in one study, people were treated with this drug for 18 months, and 88% of the patients who were treated with Gleevec showed no signs of disease progression. 94% of the patients were still alive, and normally, this is a leukemia, the disease would just keep progressing and ultimately it would lead to death. So this is an amazing, amazing kind of response for a drug, more than most drugs that are available for cancer by far. And it had very, very few side effects, because it's really only targeting this protein that's bad. It's not affecting any other protein. Okay? So only 2% of the patients in the study stopped taking the drug due to side effects. So that's also very, very remarkable. This is considered one of the major, major success stories of sort of genetics and genomics leading to a treatment. But it's very rare. Okay? And it's rare because most diseases are not caused by proteins that don't exist under any other circumstance. They're caused by proteins that are normally in your body that are doing the wrong thing. So you can imagine a protein that's sort of overactivated or not present. These are things you can't target with a drug like Gleevec, because you get all kinds of side effects if you try and knock down a protein that you need for other things. And it's basically impossible to get a mutated protein to start functioning again by adding a chemical. So while it's very promising that this kind of thing can be done, there were only a handful of examples now where that kind of rational design of a drug leads to these kind of results, because in most cases, especially for cancer, it's a very, very difficult problem to distinguish cancer tissue from normal tissue.